Accelerating Cancer Research, a Glimpse into the Complexity. The purpose of this presentation is to discuss the issue of complexity in cancer cell biology and to see what implications this may have on the topic of accelerating cancer research. With apologies, we'll begin with a possibly overly simplistic analogy. If you saw the Earth from a vast distance, you'd know something about it. You might know that it rotates around a sun, that it was about 93 million miles from that sun, that it completes a rotation in about 365 days, and you could make some guesses about the elements that might make up that planet. And if you got closer, you'd see that the Earth has an atmosphere, and that with that you would surmise that could be a balance of oxygen and a density of the atmosphere that could support organic chemistry and complex molecules. But if you really want to know what's going on that Earth, you'd have to look at the signaling system. And here's an image of a signaling system, in this case a collection of cell phone screens seen at a political protest. Now I'm going to come back to this analogy of far away, near, and close, but first share a biologic perspective. As we know, there's 23 pairs of human chromosomes, 46 in all, and that if you unraveled a chromosome, it's actually a large thread of DNA, and along that thread of DNA are segments which are what we know functionally as genes. And because there's about 20,000 protein-producing genes that drive the production of about 500,000 proteins, genes alone do not tell the story of cancer. So as an aside, you might ask, well, how do so few genes drive the production of so many different proteins? And the answer is that there's all kinds of systems called post-translational modifications or whatnot, and this, what it means is that there's lots of different biologic or biochemical reactions that happen downstream once a big protein is made that can chop it up into smaller proteins, or small proteins can be assembled into bigger proteins, and there's all sorts of slicing and dicing that can happen after the gene instructions have been used, and this explains why genes alone do not always tell the whole story of biology. In addition, there's a great multitude of factors that contribute to the development and persistence of most cancers, and because emergent behaviors result also from downstream regulatory systems, we're going to talk about in a minute epigenetics and signaling pathways. Gene profiling alone is again not a definitive cancer diagnostic treatment or prognostic tool. At another layer, there's something called epigenetics, and so toxins, chemicals, diet, drugs, and other factors can alter gene expression without changing the genes. And this is called epigenetics, and this explains why identical twins often do not get the same chronic diseases or the same cancers. And what happens with epigenetic factors is that they literally cause chemical reactions along that DNA thread, and they interfere with gene expression without changing the sequence of the gene, and so therefore there's no mutation, but there's a difference in gene expression. And this is a second layer of what I'm going to call complexity in the cancer cell biology. So as we said, epigenetics adds another layer of intrinsic complexity or functional emergence to the management of cell function. So what's going on in the rest of the cell? Of course, genes and epigenetic factors are kind of the instruction sets, but the rest of the work of the cell of reacting to the local environment and producing the proteins and all of the different parts of the cell function that need to go on, that's happening inside the rest of the cell. And question may be, well, what does that look like? So we have an illustration here that would suggest that that there are probably, without much exaggeration, there's probably trillions of molecules interacting in billions of reactions across thousands of chemical pathways with hundreds of overlapping control systems, and this is what kind of is the rest of the business of the cell as the biology of the cell proceeds. So again, apologizing for the possibly simplistic analogy, knowing something about the Earth from afar is something like knowing about the genes of a cell. It's part of the story, but not the whole story. And then if you know a little bit more about the Earth and know that it has an atmosphere and it has oxygen and supports macromolecules or whatever, then that may be something like understanding the epigenetics of a cell. And finally, if you really want to know what's going on the planet Earth, for instance, we would suggest that this, looking at the signaling system across the Earth is an important way to really know what's going on, and the same is true for the cell. And again, as we see some complexity or functional emergence, we'll say at the gene level, because one gene and one there's not a one gene, one protein relationship, and then cancer is multifactorial probably, so there's just a lot of, quote, noise at the gene level, and with epigenetics there's other kinds of complexity or emergent behavior kind of potentially observable. And what we want to do is kind of look at the third layer of complexity, the cell signaling layer, and suggest that there are just dozens and dozens of different kinds of activities that are going on here, and as such, a third layer of complexity. So I'm going to summarize for a moment. For complex cancers, if you knew all about the genes of a cancer cell, you'd know how to classify and treat cancer, right? And the answer is, well, not really, because genes don't have a one-to-one correspondence to proteins, and proteins are involved in essentially all aspects of the work of the cell, and because epigenetic changes can alter gene expression without causing a mutation. So if you knew about the epigenetic changes in a cell, would you then know how to classify and treat all the cancers? And the answer is, unfortunately, probably not, because the reaction of the cellular system, signaling system, to genetic, epigenetic, and local microenvironmental factors likely adds a third layer of complexity to the cancer cell that can be independently driving cancer mechanisms such as resistance to treatments or metastatic tendency. So in the following several slides, we'll want to present two ideas to support the concept that cancer cell signaling systems may indeed add a third independent layer of complexity, and also why this may be important as we talk about accelerating cancer research. So let's look at the topic of cell signaling and why it may add another layer of complexity in cancer cell biology. And we'll start by suggesting that most models of cell signaling systems are built with the following conceptualization. An enzyme drives a chemical reaction, like green arrow number 1 would be an enzyme, and protein A is converted into protein B by the action of that facilitating biochemical reaction, that enzyme at green arrow 1, and then another enzyme might drive a subsequent chemical reaction like green arrow number 2, which transforms protein B into protein C. And in classical kind of cell signaling modeling, the more that protein C is produced, it would have a feedback loop that would block arrow number 1, the first enzyme, and it would essentially shut down the process. And again, this is a classic feedback loop in biology. But we're going to suggest that cell signaling models today are based on the idea that each reaction or box follows its own rule, and the overall system is the sum of its parts. But we suspect that more advanced models of cellular signaling will suggest that it's a much more complex ecosystem that exhibits behavior that is far different, or potentially different, than what classic models would predict. So we're going to suggest that using overly simplistic cellular signaling models to address questions in cancer research may be similar to applying Newtonian physics to relativistic questions, and the problem is you just don't always get the right answer. So what I wanted to do is present a couple of examples that may help explain why an aggregation of apparently simple cellular signaling subcomponents creates functionally emergent behavior in cellular signaling systems. First, it's important to note that very simple systems can exhibit emergent behavior. So again, before we proceed with this analogy here, this example, I want to restate where we are. We've suggested that genes alone are not the whole story in understanding cancer biology, and that epigenetics adds a second layer of complexity, and that we think there's a third layer of complexity in the cellular signaling level where different kinds of behavior can lead to what would be functionally emergent behavior at the signaling level. So as a simple example of dynamics, we're going to consider a system that has two simple rules. The rules are stay close to your nearest two neighbors. That's the first rule. Second rule is stay away from all others. And what we're going to show in this next slide is a modeling software that can throw 30 random points onto a computer screen as a blob of points, and each of those points will follow an instruction, which again is those two instructions: stay close to your two nearest neighbors and stay away from all others. And then we'll see what happens in that ecosystem, again as an example of the dynamics that can come from an ecosystem that is unexpected. So here we go. I'm going to punch go here, and what you'll see is 30 dots that are randomly thrown onto this computer screen, and at 15 times a second each point has an algorithm that says stay closest to your nearest neighbors, to your two nearest neighbors, and stay away from all others. And that is the only instruction set that's there. And the point of the exercise is to show that the dynamics here create another layer of structure, which is obviously a circle if we let this keep running, and that what appears to be a very simple set of rules and simple dynamics, somewhat analogous to that first slide of A goes to B goes to C with those two enzymes. And you might think the feedback loop, and then this is just an example that all those feedback loops likely could have emergent kind of behavior through very simple dynamics such as this. So in cellular signaling systems with thousands of reactions that simultaneously share dozens of components with overlapping control system and feedback loops, it's not surprising that unexpected behavior may emerge. And if you concentrate just on one set of reactions and one kind of therapy kind of in that ecosystem and thinking that'll characterize an output or outcome, it just turns out you'll often be wrong, and you won't understand maybe why that you are wrong. And so this may explain in part the unpredictable resistance that is seen in response to some targeted therapies in cancer that are coming forth. Again, the idea being that our simple understanding of a pathway, a signaling system pathway, may just be overly simplistic, and there may be simple dynamics that are emerging that we just don't know to see or hear. Another aspect of cellular chemistry that may drive complexity in cellular signaling is explained by fast-slow reactions that share common components. And this whole line of conversation evolves from the work that Alan Turing did in the 40s. Alan Turing was a man famous for cryptology and decoding Enigma and computer science and artificial intelligence and mathematical biology. And in his work in the late 40s, he predicted the existence of oscillating chemical reactions. And these oscillating chemical reactions were actually demonstrated 10 years after he predicted them, and so that's an interesting dynamic that may be important in cellular physiology. So let's look at that. In classical thermodynamics, hot plus cold equals warm, so hot water plus cold water would equal warm water. And you can say conceptually, if combining hot and cold water resulted in an oscillating chemical reaction, the result might be that a glass of water would alternate between hot and cold, and that would make no sense to us, of course. But that may be sort of the way to think about, or what you see in some of these kinds of reactions that he was talking about. So thankfully, again, the combination of hot and cold water does indeed result in warm water, but the following slides demonstrate an oscillating chemical reaction, which is a series of parallel fast and slow chemical reactions that share common constituents in different parts of the pathway. And again, the point here is not to understand the chemistry, but just to simply understand that this may be an analogy to cellular signaling systems. And what happens here in this example is that there's these three solutions which can be combined together, and this is what Turing's prediction was. These are solutions which, when put together, create fast, slow reactions with common intermediaries. And again, the chemistry is not important, but when I start the reaction, you can see that it varies back and forth. And the point here is that the oscillating reaction is produced by this combination of reactions which are fast and slow and which have common molecules that go between the two. And so the point is not that exactly this specific reaction is happening or anything like that, but the point is that's likely that self-organizing tendencies, as we saw in that computer modulation in the slides before, or the oscillations in chemical reactions across time and reaction scale may meaningfully contribute to the functional complexity of the cancer cell signaling ecosystem, and that traditional models of cellular system signaling are vastly oversimplified, and thus that new tools and approaches are needed to address the issue of complexity in cellular signaling. And again, we think this is kind of a third independent area of complexity or emergence that may be part of the cancer cell. Adding to the complexity of the cellular signaling story is the tumor microenvironment. So again, as we showed in the previous slides, there are simple dynamics like the computer simulation, which may suggest that there could be kind of emergent kind of behaviors that we wouldn't necessarily suspect in a simple system. And the second example of the changing chemical reaction just suggests that there, again, it could be other kinds of dynamics which would make the simple model of cellular signaling not an appropriate or effective model. And then thirdly, we know that the tumor microenvironment has a significant impact on cancer cellular signaling by upregulating or downregulating various microenvironment-sensitive pathways. So this is the microenvironment is also a non-trivial third mechanism, and I'm sure there are others which impact essentially the functional emergence of the cellular signaling level. And so again, a future vision or a future possibility is that more advanced study of cancer cell signaling systems may reveal signaling response patterns or families of patterns that result from exposure of the cell to cancer treatments, both targeted and classical treatments. And these patterns will likely teach us that treatment of complex cancers will consistently fail with isolated targeted therapy directed at a specific cell line, or directed specifically at a genomically heterogeneous tumor mass or a progressively heterogeneous metastatic lesion. And it may also suggest that successful treatment of complex cancers requires sequenced and combinatorial targeted therapies in order to drive cellular systems into reactive zones where resistance to treatment and alternative pathways cannot prevail. And we're going to look more about this in subsequent slides. So again, in the following slides, we'll use the analogy of neighborhood streets to present the idea of cancer pathways and how targeted therapy may block part of a pathway, and how resistance to treatment may emerge, and why sequenced combinatorial targeted therapy may be the essential step or an essential step in the effective treatment of complex cancers. So again, apologizing for this very crude analogy, we'll surmise that a cancer cell becomes or is a cancer because it has an active abnormal pathway that results in uncontrolled cell growth. And in this analogy, point A, getting from point A to point B, requires traversing the series of little dots that go from point A blue to point B blue. And getting to point B causes uncontrolled cell growth in this crude analogy. And so again, we're looking at the idea of a cancer signaling pathway starting point and an ending point where the end point drives uncontrolled cell growth. And we'll see that in this little analogy, there's lots of potential targets for targeted therapy. And you can imagine that the problem is that when you block one step of the pathway, you might have some effective treatment response for some time, but that other paths could come forth and sort of make that initial targeted therapy ineffective. And there are targeted therapies that have come forth that have very, very good efficacy and effective treatment that last for a few months and then lose their effectiveness. And conceptually, this is likely what's going on. And you could block, again, not just one pathway, but a series of pathways, of series of steps of the pathway, and still another path of resistance could emerge. Resistance being defined as a regrowth of the tumor or metastatic growth. Thus, we're suggesting the very simple idea, and this is not an original idea, that effective targeted therapy to treat cancer may require a sequence of targeted therapies that drives a cancer cell's signaling system to a sensitive point, and that's what we're going to illustrate here. So you can imagine that the treatment of a complex cancer might begin with 2 targeted therapies, and that limits the options. You can see kind of, in box 1 and box 2, that might be 2 different targeted therapy drugs, and that would leave, the path from first blue dot to the second blue dot. There could be an alternative, which could, as in that red illustration, could evolve treatment, and then there could be addition of subsequent targeted therapies 3 and 4, which again would leave a path, but would be narrowing the options as this system would through all kinds of different mechanisms find alternative pathways to meet that same end, and subsequently, again, this is not to be too trivial here, but another set of targeted therapies 5, 6, and 7 could drive the path down, finally to have no choice but to get to this kind of uncontrolled cell growth, except to be coming along as the arrow shows. And then, of course, targeted therapy number 8 would end the game. And so this would be potentially an idea about how we'd evolve in targeted therapy, and we're suggesting that, advancing cancer research progress may require us to think a lot more about sequential targeted therapy. And again, the people are out there doing that, but I just highlight that, and the sequencing is important. And again, the idea is that if you just started with that number 8, the last in the sequence, and said we'll block that part, what you would see is that there's alternatives to get to uncontrolled cell growth. So it may just be that our isolated targeted therapy kind of era of today will be a bygone era, and when we get to effective cancer, complex cancer treatment in the future. And obviously these are all potentials for targeted therapy, and that the future of targeted therapy probably includes not blocking, only the nodes involved directly in the tumor progression, but the surrounding nodes that account for resistance. And as we said, people are working in this area, and there's publications. This one's targeted therapy for melanoma, rational combinatorial approaches. This is a beginning of the same discussion. Directed phenotype switching is an effective anti-melanoma strategy. This uses a combination of a traditional therapeutic agent, which drives changes in the cellular signaling level, and then the combination of that with a targeted therapy has yielded some good results. And then finally, this uses both the word sequential treatment strategies and combination therapy regimens in metastatic renal cell carcinoma. So this is out there, and again, our commentary is that we think these are absolutely the direction of the future. And again, that studies in sequencing and combining cancer therapies are not new. The sequence and combination of cancer radiation and surgery and traditional chemotherapy have been studied for decades. However, studies in sequential and combinatorial targeted cancer therapies are a more recent development, mostly because the array of targeted therapies is still developing. And really, a primary purpose of this communication seeks to suggest that accelerating the effectiveness of cancer research at this time requires not only the study of sequence and combination of targeted therapies, but also characterization of the functional emergence of the signaling layer, which is, again, a key part of this discussion. So with that, we will conclude with 10 points which summarize that which we've said. First, knowing about the genes and mutations of a cancer cell doesn't necessarily tell you everything you need to know about the diagnosis, treatment, or prognosis for most complex cancers. Second, the existence of epigenetics, which are non-mutational factors that change gene expression, means that gene sequencing alone does not tell the whole story in cell biology, and that epigenetic factors likely add a second layer to the complexity of the ecosystem of a cancer cell, and that cancer cell signaling systems likely have inherent complexity, adding a third level of complexity to the cancer cell, and exhibit emergent behavior that is amplified by interaction with the local microenvironment of the tumor. And that number 4, accelerating progress in cancer research requires an appreciation that there are 3 independent layers of functional complexity and that emerge at the gene, epigenetic, and signaling layers of cancer cells. And some of the inherent complexity of cellular signals may be due to emergence that's related to simple dynamics, A, B, overlapping components among different systems, kind of B, C, the oscillating reactions we saw an example of, D, redundant pathways and feedback loops, and E, other mechanisms. 6, in complex cancers, understanding diagnosis, treatment, Treatment resistance, which is recurrence or metastasis, and prognosis may require understanding emergence at the final layer of cancer regulatory mechanisms, which is the cellular signaling level. And it's likely, and that may be the most important level, I guess is what we'd say. It's likely that cancer cell response to treatments, especially targeted therapy, will be characterized by patterns or families of patterns of changes to cell signaling pathways that are directly associated with treatment failure, resistance, and/or metastatic tendency. 8. Heterogeneity of cancer cell lines within a tumor across microenvironments and across metastatic lesions represent additional dimensions of the challenge to deal with the emergence in cancer cell signaling. Effective treatment of complex cancer likely requires combinations of targeted therapy used in a defined sequence, and accelerating progress in this area likely requires new research platforms directed at computational modeling to characterize the emergent aspects of cancer cell signaling systems, because we'll end up wanting to do this, we think, in silico, as in the computing realm, rather than trying to do the volume of experiments in the lab that are required to accomplish the same thing. That's the last slide. We appreciate your attention and look forward to any questions you may have.